All right, so I'm going to go forward, and this is going to be the lecture on the remaining parts of the physiology lecture. So, so up through now, what we have discussed in physiology is how chemotrophs, both chemoheterotrophs and chemoautotrophs, um, go through different processes, use carbon sources from organic matter in order to drive glycolysis, the TCA cycle, and if they have the components and terminal electron acceptors, the electron transport chain. So now I want to kind of shift gears here. I'm going to talk a little bit about the photoautotrophs, or the photosynthetic capable bacteria. And these bacteria predominantly come in the form of cyanobacteria. And you find cyanobacteria in a lot of different places, but one of the most common places you find them are in the ocean and in waterways. So these are green bacteria, right? And so if you go back to the first lecture, we talked about the green photosynthetic bacteria versus the purple sulfur bacteria, and how they kind of coexist or co-symbiotic in certain environments. And so these bacteria basically perform photosynthesis. So they have found a way to take sunlight, right? So they take sunlight and energy from sunlight, and they activate um, chlorophyll. And chlorophyll are basically small membrane brown green pigments that form these different structures called thylakoids. So thylakoids are basically a collection of chlorophyll or chloroplasts that form these three-dimensional structures. So if you're talking about chloroplasts inside of plants and algae and other types of photosynthetic organisms, they kind of look like this. And so they're double membrane bound organelle, and inside of them they have these discs of chloroplasts, discs of chlorophyll. And these discs of chlorophyll form what is known as the stroma. And these stroma come together in these even bigger stacks and create what is known as thylakoids. So thylakoids in plants are very much vertical and linear, and they form these stacks, almost like a stack of pancakes. The thylakoids so the chlorophyll and the thylakoids of a cyanobacteria kind of looks like this. So basically, they are stretched out, so they're elongated along a horizontal plane. And how these work, how these random photosynthetic components work, is that they can absorb sunlight inside the green pigment. Again, pigments are really important for absorbing UV radiation and electromagnetic radiation in the form of energy. And it allows for what is known as the light dependent reactions. Light dependent reactions effectively create ATP for energy, and they also create electron transporters, NADPH and NADH. So remember, these exist as NADP plus, right, and NADH plus, also NAD plus. And so when they are affixed to a hydrogen, these are hydrogen atoms, right? Not hydrogen protons, they're hydrogen atoms. So that added electron gives it a neutral charge. So these components that are created during the light reactions are sent over to basically fix carbon dioxide, or convert carbon dioxide. So these organisms have ways to absorb carbon dioxide. So in plants, this is in the form of the stomata, right? So the stomata, they open at night. They allow for carbon dioxide to occur, or also known as the dark reactions, because they open at night. Carbon dioxide is filtered in, and with the help of enzymes and ATP to drive the reactions of those enzymes and electron carriers, we can then generate organic molecules such as glucose. And then glucose plus ADP and the electron carriers are then sent back to the light reactions in order to drive glycolysis and the remaining other functions of glucose, right? So everything from central metabolism to generate ATP, as well as DNA, amino acids, etc. So inside of the actual membranes of these photosynthetic bacteria, and we look inside of the ability to drive photosynthesis. This happens through a series of enzymes and enzyme complexes known as photosystems. So these photosystems are what absorb the light energy, and they basically use the electrical energy that is generated from that light energy to then drive the function of what we call reaction centers. So if you follow this kind of chart over here, or this legend over here, the light harvesting complexes, right? These are the absorptive molecules. These are the pigments. They drive and move energy amongst all of them to get to a reaction center. And when this happens, the reaction center is able to decouple the electromagnetic energy from the electrons and photons that make up those electromagnetic spectral waves. So the energy released is allowed to make a proton motive force. So we have water, right? Most living things have water. We are able to decouple water, create those hydrogen atoms. Hydrogen atoms, again, allowed for the creation of the proton motive force. The proton motive force is what is used to drive ATP synthesis, like a chemoosmotic reaction like we've seen before. And then the electrons are decoupled and allowed to bind to NADP plus or NAD plus, forming the neutral charge NADPH and NADH species. So if we're looking at this from an oxygenic standpoint, right? So if we have, again, going back to if we have all the components, so if we have access to oxygen, carbon dioxide, usually coupled to water, again, because we need to split water to generate the hydrogen protons and the hydrogen atoms, right? So we have CO2 and water and the light energy being decoupled and generating electron carriers in the reaction centers, allows for the creation of the fixation of carbon dioxide into glucose, because we're using those carbons to generate glucose. The result is, again, because it's 2 oxygens per carbon dioxide molecule, right? And we have oxygen from water. We use the oxygen from water to drive dehydration synthesis to decouple carbon dioxide and then to create glucose. The extra oxygens from carbon dioxide is then left as oxygen because, again, we have no more carbon that's been made into glucose. And then the remaining water that's unused remains to then be cycled back into the system to drive again. Because, again, we have how you've used half the water, so 12 water to 6 water. So we get 6 more water, and we can continuously cycle this system, which is important because you're trying to balance between light and day reactions, right? If there's no oxygen available, right? Again, we still start with carbon dioxide, we still have light energy, but we still need an electron donor, right? We still need a way to get electrons from the hydrogens in order to generate our electron carrier species. So in this case, this is 2 hydrogen, and then A meaning that it has to have some type of anion. So this could be a hydrogen sulfide, it could be hydrogen phosphorus, any of those group 7, group 8 elements can be used here as long as it has a negative charge. And then, of course, again, carbon dioxide can then be fixed. You're left with the anion as a byproduct, and then water left over. So if you're looking at these systems, right, of course, plants and other green photosynthetic organisms like algae, they all use oxygenic photosynthesis. They require to do this. Obviously, plants, they are exposed to carbon dioxide and oxygen out in the environment because they give off that oxygen. Algae do the same thing. So antioxygenic photosynthesis are really for deep water level eukaryotic photosynthetic organisms, but more appropriately, it really is only found in photosynthetic bacteria. So when we look at these particular light systems, right, these photosystems, they are effectively just like the electron transport chain. So they exist in the membrane, and how light passes through and energy passes through these systems is actually kind of inverse of what we think. So the first photosystem or antenna pigment that receives light energy is photosystem 2. And photosystem 2 again creates or decouples, like I said, decouples water, creating oxygen species and hydrogen protons. And this releases those electrons in the form of just actual electrons, where and so the electrons are received by photosystem 2. They're then sent through a series of cytochromes, just like we have in a chemoheterotropes and chemoautotrophs, right? So we have cytochromes or electron, intramembrane electron carriers. They move the electrons through to photosystem 1, which also can use light reception. So if we're following down here again, our electron acceptor is the cytochrome. We have cyclic photosynthesis, the electron acceptor being another cytochrome, which also can receive electrons from photosystem 1. So photosystem 1, the energy is kind of like reset, right? So that's why we call it cyclic photosynthesis, because it takes energy to move that electron along the membrane. When it gets to photosystem 1, there's not enough energy to then reset the electron past photosystem 1. So in this case, the light energy received by photosystem 1 is to decouple energy from electromagnetic UV radiation, and then that gives it enough power to send that electron through to another to another cytochrome. And then finally, at the end of this, the electron actually is then sent to NADP plus reductase, or NAD plus reductase, which then takes NADP plus or NAD plus, and it couples it to this high, again, because we're making this chemoosmotic proton motive force. So we have hydrogen protons. Those hydrogen protons are then coupled to that electron, creating hydrogen species, hydrogen atoms, where they can then be coupled to NADPH or NADH. So throughout this system, using these electrons as they move, so the second part of this is the generation of ATP, right? So as the hydrogens themselves, again, because we're splitting water, right? So we have hydrogens inside the cell. We have other hydrogens on the outside of the cell, just from kind of this generation of a chemiosmotic imbalance. So these hydrogens that are created intracellularly are sent to ATP synthase, just like we have with chemoheterotrophs. The difference here is instead of creating an extracellular hydrogen gradient, we're creating an intracellular hydrogen gradient. So this is inside the cell. So this is inside the cell. So where hydrogen builds up on the inside of the cell, it then wants to move, it wants to diffuse outside of the cell because it's charged, it can't. So this is where ATP synthase, and you can see here ATP synthase is actually turned upside down, right? So the the actual catalytic component is facing the outside part of the cell, right? Or outside part of at least the thylakoid, excuse me, not the cell, but the thylakoid. So this is actually I take this back. This is inside the thylakoid, not the cell. Inside the thylakoid. Sorry. So the whole thing is still inside the cell, right? Because it makes no sense to let ATP escape outside the cell. I don't know why I was thinking that. Sorry. So inside, again, we're inside the stroma, right? We now need to get hydrogens. We have lots of hydrogens inside inside the thylakoid. We need to push them back outside the thylakoid. So we now are pushing hydrogens out, driving that mechanical to chemical energy transversion of ADP to ATP. And the cool thing is, by doing this, we now are creating the situation where we have hydrogen. We're creating a new gradient, right? We're kind of establishing an equilibrium and then pushing past that equilibrium to generate another chemiosmotic imbalance, hydrogen being on the outside of the thylakoid, not inside the thylakoid, where these hydrogens can now either end up here by creating electron carrier species or come back to the electron transport chain and be shuttled back across the membrane. And this is where we get the sense of cyclic photosynthesis, right? So, if we kind of follow this along, right? Oxidative photosynthesis, same thing, generates 36 to 38 ATP per molecule glucose, or in some essence, per molecule carbon dioxide that is converted then into glucose. Whereas anoxic only creates 4 to 6 ATP per. And, just like plants, right? So, I don't want you to be lost on this, but just like plants, there are light and dark reactions here, right? So, so it is important to note that, that and you have to think about it from the perspective of what makes sense, right? So the light reactions are really in order to generate the conditions in order to drive synthesis of glucose. And so the dark reactions are what really occur when you create glucose and then the subsequent metabolism of glucose. Now, the cool thing is in plants, because of the ability to lose or gain water, especially because of the evaporative nature of water, this is really kind of classically defined as light reactions that generate electron transporters and and basically fix carbon dioxide in order to generate glucose and all the functions, because the nighttime reactions, obviously, you're not doing light reactions during night because there is no light, right? So the light reactions are to generate kind of the byproducts in order to generate glucose, and then the night reactions are where you get absorption of carbon dioxide, and because you have all these precursors already pre-made, absorbing carbon dioxide can then quickly be fixed to glucose and then run glycolysis and all the other glycolytic cycles. For bacteria, because there's no, per se, absorption or loss of water, and they don't have stomata to kind of generate carbon dioxide fluctuations, whenever they get access to carbon dioxide, whether it's light or day, does not really matter. They can fix carbon dioxide kind of at will. They do kind of adhere to light and dark reactions, but the dependency on when those reactions occur, unlike plants, is a little less, or I should say, is a little bit more fluid. All right, so we talked about chemoheterotrophy and we've talked about photoautotrophy. So the next important question as we move forward is, and this is kind of a really complicated question, which is what is energy? What exactly is energy? So in physics, right, we define energy, or in science in general, we define energy as the ability to do work. It is the underlying current or function in order to do work. But obviously, we're not really talking about physical work when we talk about biology. I mean, we can do work, but as physics defines work, it's very different by how biology defines work. Because in physics, to define work, there is a certain directionality of movement and force that's applied for that movement. Right, go back to the old equation F equals ma. But in biology, this is not necessarily as true, because in biology, sure, we can have movement, right? Humans move and other organisms move, and even bacteria move, but maybe not in the universal directionality that is required for work. And beyond that, um, we know that work is differentially defined chemically, right? So in order to do a job, for example, in order to perform a certain function, is really kind of what we're talking about when we're talking about biology or chemistry. And so when we're talking about functionality, when we're talking about actually doing something, what we're really talking about, therefore, is enzymes, right? Proteins and enzymes, because they are the only of the macromolecules, or of the submolecular element or subcellular elements that actually do functional work, right? You talk about DNA doesn't really do work, right? Has work acted upon it. Same thing with lipids. Lipids are synthesized and they exist and they do a job, but their job is more chemically and structurally defined, not because they actually have moving pieces. And same thing with with sugars, right? Sugars are crystalline, so they don't, and they are the most rigid of all these things. So what we're left with is enzymes, right? And so as we move forward, we talk about the generation of ATP and all these metabolic functions. Like I said, central metabolism is really for survival, and how we think about it from a molecular Darwinian sense. But in reality, that if you would you peel off that kind of top layer of Darwinian or molecular Darwinian survival, what you'll find is what drives the ability for all the enzymes to work, and ideally to work optimally, is the ability to get access to all of those resources to build those enzymes, and then for them to function, right? And so that's really the key of enzymes. So going back to all those things, right? Survival, reproduction, all of those things. To survive, you need enzymes to function, because again, we exist in a positive delta G existence, meaning that we don't have spontaneous reactions that occur, and we need to live long enough to reproduce, right? And that's the definition of Darwinian fitness, and why we need to be fit enough to challenge for resources in a particular niche. All of that really boils down to getting resources for all of those central metabolism functions that we talked about, and those central metabolism functions to generate ATP. Why? Because ATP is required for enzymes to function. So, so when we think about enzymes, here's kind of the general anatomy of an enzyme. Enzymes are by and large biological catalysts that, again, lower the energy barrier, or the energy threshold for a chemical reaction to occur, because they themselves can perform chemical reactions. So their existence allows them to perform chemical reactions. And enzymes can be either truly organic, meaning that they are only made up of elements that form or amino acids that form certain kinds of bonds that do not have any non transition metals or group 1, group 2 metals in them, or they can be metalloorganic, meaning that they have a metal cofactor or they require a metal substrate in order to perform their catalysis. And so enzymes typically have a body, and the body is just how the protein folds around the particular uh, polypeptide chain that is created. And that folding eventually creates what is known as active sites. Now, active sites serve kind of two really important functions. The first is that this is where the enzymatic biochemistry occurs. So, if an enzyme is involved in a chemical reaction, this is where the chemical reaction occurs. The second thing is that the folding, again, to go back to thinking about what we talked about, right? Protein structure equates to protein function, which means that the amino acids, right? The amino acids in the active site actually matter because those amino acids have to be able to be capable of performing that chemical reaction, meaning that they have to either be able to bind a metal to perform a reaction, or they themselves, their R groups or the bonds that they form, have to be able to move atoms or electrons or other types of atomic components around to rearrange ultrastructures of chemicals or molecules. Um, so if function is dictated by structure, the structure therefore is very specific, which means that the things that can bind to the active site, right, what we call ligands, or the molecules that bind to an enzyme, are specific to the enzyme. Or in other words, an enzyme is very specific to its ligands. So there are two kind of models for enzyme substrate or enzyme ligand interaction. There's the lock and key model, and this is the kind of general sense of what we think of when we talk about enzymes, and that there is an active site. Again, the active site has a specific shape to it, and I want you to kind of understand this, that as the substrate binds or the ligand binds, it fits in the grooves of that particular enzyme. And then what ends up coming from the substrate of the ligand, the end product, is just called that. They're called products. So we start with substrates or ligands, we end with products. And if you think about it, you might be thinking, okay, well, if there's chemistry being done and you have this really tight binding from an enzyme and a substrate, how do the products get released? Well, if you think about it, products that you create are chemically distinct from the substrate, right? These two products are very different shape than this one substrate. So if enzymes are specific to their substrate or their ligand, once you change that substrate in the ligand, you change that affinity, you change that ability to bind. And so, this is going to be kind of a little bit warped, but there's also another kind of fit, and this is called induced fit. And this is down here, where we have a particular substrate, and the substrate itself, when it binds to the enzyme, it does change the enzyme shape, and this change in shape distorts the bonds between the enzyme and its active site and the bonds between the substrate. And this allows for the substrate to be catalytically converted from the enzyme, creating the same general products. Now, induced fit doesn't happen often because, again, substrates are particular to their enzymes, and enzymes are specific to their substrates. However, if you think about it from a kind of global perspective, and this is something we've talked about, right? When you have an enzyme, anything that, this enzyme, its shape is solely based on the environment and the sequence of amino acids and the polypeptide chains and how they all fold over the four types of folding, right? The four levels of folding that a protein undergoes. And so anything that we add, including like even something simple as a phosphate group, can change the shape. It might not be It might not be a massive change, right? But anything that affects the chemical makeup, the environmental makeup, the folding pattern of a protein is going to change the shape of that protein, right? So basically, if anything interacts with the protein that's different from how it was originally created, the protein changes shape. What that change equates to, it really depends on a case. It literally depends on a case by case basis, but it always does change the shape. So again, even here, even in the lock and key model, this binding of the substrate is going to very subtly change the overall structure of the protein because you are changing the environment of the protein. The protein wasn't bound to substrate before, and now it is. So this is why induced model actually does fit because, for example, if this part of the substrate binds first, that is going to induce a change in shape, and then that change in shape might move this second component closer or turn it, and therefore creating a new kind of active site shape in order to best fit the substrate. So enzymes typically exist in one of two forms. When they form their globular structure, they can either come off the ribosome and fold in their four levels of folding and be an active enzyme, and those are just called holoenzymes or complete enzymes. And then there are those that are called apoenzymes, and these are enzymes that are created. They go through their four stages of folding, and they are not active. And so when they exist in the non-active form, because they then need to bind something, either a coenzyme, another enzyme, they need to bind a metallic cofactor, they need to bind another substrate or a phosphate group to activate. They need to bind something to activate them. Now, whether or not an enzyme either needs to be activated or is active off of the ribosome really depends, again, on a case by case basis. But by and large, enzymes that are required for enzymes that are required for um, enzymes that are required for um, your day-to-day tasks, right? So things like ATP synthase or all the enzymes in glycolysis, all of those enzymes probably exist in the active form because whenever you need, in that example, whenever you get glucose, you want to be able to convert it in central metabolism or at least get it in the central metabolic pathway and then cut it out to catabolic reactions or anabolic reactions if those other molecules such as nucleotides or lipids or amino acids are required, right? So on the other hand, if you have enzymes that you might need that might be important, right, but that are not essential for just everyday just survival, if you're a bacteria, you may produce enzyme, and your cells do this too. You may produce proteins, you may produce enzymes that are at the ready, right? They exist at the ready, but they exist in an inactive form. And so basically, you have enough energy to create those proteins, and at a very basal level, not at a very high level or very low level, at just a base level, you produce some of those proteins. And usually these are proteins that can be inducible, right? These are proteins that you can make more of at a moment's notice. So you have some to start with as you start creating more of them. So if it's something that can exist because you have enough energy to create it, but you don't need it active right now, those are typically these inducible enzymes or able enzymes. So, and not to be kind of lost out on this, okay, right? Normally, when we say that enzymes are specific to their substrate, normally it's one enzyme to one substrate, right? And this is, probably 80% of the cases. But there are a few examples in the world that some enzymes can actually fit multiple substrates. And typically, multiple substrate enzymes require all of the substrates in order to perform their catalysis, whatever that is. So if we have in this example, right, we have substrate 2 and substrate 1. Typically, one substrate, in this case, substrate 2, has bound first. This allows, again, so substrate 2 binds, right? There's a conformational change. So we'll just say delta. This results in the creation of a new or open active site. This is now the right fit, lock and key fit for a second substrate, in this case, substrate 1. Once both substrates are in, right, then these can be chemically converted to their products. And both substrates are converted at the same time, typically because one is required in order to allow for the second one to bind, and then the use of ATP is then more efficient because now we have one ATP molecule to drive the creation of products from two different substrates. Now, if you're thinking, why then do most enzymes only have a single substrate affinity, and some only very few have multiple? And this if you think about it evolutionarily, right, this makes sense because you can't always depend on having all the substrates for a particular 2 or 3 or multiple substrate enzyme. And if you need one of those products as an essential product, and one of them might be an important but not essential product, unless you have both of those initial substrates, you're missing an essential product. Whereas if you have an enzyme dedicated to the functional creation of another molecule, you don't need to depend on another molecule to bind, right? So not kind of pigeonholing yourself to requiring two substrates is why. So yes, where it is more efficient for energy usage, it's not biologically um, it's not a biologically fit system that all your enzymes should fit multiple substrates, because without all of those substrates, think about it again from a resource competition standpoint. Without having all of those substrates, the multiple substrate enzymes can't function, right? So enzymes, they exist really kind of in two superclasses. There are exoenzymes, and we talked about this in lab. Exoenzymes are enzymes that are, again, created inside the cell. They're then exported outside the cell, and this export does kind of activate them. So this is an example of an apoenzyme that turns into a holoenzyme. We create apoenzymes that are inactive, and then they're shuttled outside. And once they are shuttled outside the cell, the outside environment, again, because it's a different environment, intracellular environment, extracellular environment, this again causes a change in shape to the enzymes, and something has to happen, whether it's a change in acidity, change in molecules, or phosphorylation, right? Something happens to these enzymes outside of the cell, some activating reaction occurs, and this activates the enzymes, allowing them to then bind their substrates and chemically convert them. The other subclass or superclass of enzymes are called endoenzymes. These are ones that are born and remain inside the cell. These can, unlike exoenzymes, which are always inactive inside the cell and only active on the outside of the cell, endoenzymes can be created either as active, right, or they can be created as apoenzymes and remain inactive until they are activated through another function. So once we talk about the creation of enzymes and what they do and how they kind of generally function, now I want to talk to you about kind of the different chemical reactions that typically occur in enzymes. So there are two really important ones, and these are probably the two most common, or universally conserved, and found in 50 to 80% of all enzymes, because these are just general reactions that can do a wide concatenation of different reactions. And these are the condensation reactions and hydrolysis reactions. So condensation reactions are in effect the way to take two substrates or two or more substrates, And these are typically anabolic reactions, right? So we're taking two separate substrates, and then we dehydrate, condense water through the function of these enzymes. And by condensing water, we create a new stereobound bond. And this bond allows the creation of a new, larger molecule. So these are typically called condensation synthesis or dehydration synthesis reactions because we're typically synthesizing a larger molecule from two or more smaller molecules. Um, One of the probably the most common, and this is what you find in all of the a lot of the central metabolism reactions, right, is hydrolysis. So if condensation reaction is creating water because we're condensing basically hydrogens and oxygens to create water, hydrolysis, as the name suggests, hydro, water, lysis, breaking. This is breaking molecules into smaller substrates by by basically using water. So in this case, we're using water in order to infiltrate common bonds between two molecules. This creates a breakage where a hydroxyl group and a hydrogen typically can then separate the bonds between two groups, and therefore there's no longer that linkage, creating now two separate products from a single standpoint. So if you want to take this from a central metabolism kind of perspective, right, the subconstituents of carbon dioxide, which are then created into like C3 molecules, those C3 molecules can then be bound together to create C6. Um, right, and so the condensation reactions are typically used to synthesize larger molecules or converting glucose into glycogen, because we're then combining glucose molecules by dehydrating it. Whereas when we're, say, taking glycogen into glucose or starch into disaccharides, we would cleave off a piece of those sugars by breaking the bonds between the lattice structure and a single hexose sugar in order to then generate or drive glycolysis. So that's kind of a general, like, two very moderate examples from the same system. Um, but the overall, these are the two big reactions. Um, Obviously, there are more chemical reactions than just hydrolysis and synthesis. Some other reactions that the enzymes undergo. There are redox reactions, right? So these are in some essence either reducing or oxidizing molecules. So in this case, we're not breaking or we're not synthesizing anything. We're just chemically converting things. One of the biggest classes of this is oxidoreductases. So oxidoreductases, one of the biggest examples of this creates is catalase. So catalase converts hydrogen peroxide, and it reduces hydrogen peroxide to hydrogen and half diatomic oxygen, um, in which another oxygen molecule. So if you have two H2O2, you can then create something like this. You have two waters and then an O2. So you create oxygen and water. You haven't broken anything. You haven't synthesized anything. You've just reduced, or so that's a reduction reaction. You can't have an oxidation reaction, right? Converting iron to iron oxide is an example. We're just kind of moving an oxygen over, but we don't have these massive molecules that we're doing anything with. There are transferases. Transferases move things from one to another, right? One of the biggest examples of this are phosphotransphorases or kinases, right? So, an enzyme that has a phosphate group that then moves it to another enzyme. And so again, we're not creating something brand new or breaking something. We're just moving a chemical group from one place to another. Double replacement, this is typically acid-base chemistry. Decomposition, these are hydrolysis reactions, synthesis reactions we talked about. So the last one we haven't talked about are isomerases. This is basically taking a molecule and then converting it to a different isomer or different different shape. And so, converting, say, glucose to fructose, right? They're both monosaccharides. They just have different bonds, different places. So isomerization enzymes basically just change the shape, but no change in chemical composition of a molecule, right? So we're creating isomers. That's where the name comes from, isomerization. All right, so enzymes, like I said before, you have two really kind of different types of enzymes in the way that they are folded and active. There are constitutive enzymes. So constitutive enzymes are enzymes that are basically always on, and the amount they have and kind of the amount that you produce in the sorry, so the amount of enzyme you produce and the amount of activity those enzymes have never change, right? It never changes whether or not you have or don't have substrate. If you add more substrate or less substrate, you still have the same number of enzymes, and there's no added or different amounts of that enzyme. And we're talking about constitutive and or regulated. We're talking about this at a DNA level, right? So this is expression, so number of enzyme molecules. So constitutively active enzymes or constitutively regulated enzymes are ones that are always just at a constant number of enzymes from a DNA, RNA to protein level. The other type we have are induced and repressed or regulated enzymes, right? So these are regulatable enzymes. And some induced enzymes, and I hate talking about them from induced and repressed. Let's just say that they're regulated, because if you're inducible, right, and you create more, so if the enzyme is induced, this typically is that, again, because you have more substrate, and the more substrate feeds back somehow to the DNA. So the cell knows that there's more substrate, so that it tells the DNA to make more RNA, make more protein. So you get more enzymes to deal with the amount of substrate, so your enzymes aren't fit to saturation. And at the same time, as the enzyme performs its function, as the amount of substrate is reduced, again, the cell then tells the DNA to make less RNA, makes less protein, and then you're left with less enzyme that exists just kind of at the ready, but in less numbers, because you're not going to spend energy on enzyme that you don't have. So I like to call these regulatable. A lot of biology, and I think even in the textbook, they call them inducible or repressible enzymes. But in reality, any enzyme can be induced and repressed, right? Because you can definitely say, well, at an inducible enzyme, once it has dealt with all of the substrate, you're not going to keep, you've induced it to be at a higher level. It did initially exist in a repressed state. So anything less than this, right, is going to be repressed. So let's just call them regulatable instead of inducible and repressed, because any gene can be induced and repressed if it is under regulation. So that's at the DNA level, right? but I want you to understand that again, enzymes, the amount of enzyme can be induced or repressed, so that's regulatable. But then when the enzymes are created, right? So when the enzymes are actually made, um, again, if they're made and they're constitutively active, right? If they're just made and they're active, then they're not regulated. Um, how do I put this? All right, so let me back up. Sorry. So let me pull up the whiteboard. So if you have enzymes, right, at the DNA level, they can either be constitutively active, uh, regulated, meaning constitutively expressed, excuse me, meaning that there's no difference in expression or number of enzymes, or you can have regulated, which means that they can be either induced or expression, more enzyme proteins, or they can be repressed, less expression and less enzymes. Okay, I hope you're with me so far. And then once this happens, right? So this is this is what we would call this is what we would call transcriptional regulation, right? So I'll just write it. So this is transcription regulation. So or gene regulation, right? And then we have what we call post translational regulation. And post translational regulation is we have a certain number of enzymes, right? And they exist here, and they can come in two forms. So you can have uh, active enzymes, right? So active holoenzymes. So these are the enzymes that are just created. So right off translation, they're ready to go, they're ready to do work. And then you can have inducible activation, and these are the APO enzymes, right? So these are enzymes that are inactive after translation, but then are activated by some modification after translation. Okay. So obviously all four of these things can intersect. So you can have enzymes that are constitutively expressed, but those constitutive expressions can either be active or inducible. And same thing where you can have regulated at the genetic level, but then once you do express these from transcription and translation, these can exist either as active enzymes or they can be doubly regulated. They can be regulated at the DNA level to make RNA, make protein. The protein that comes out is an APO enzyme that then needs to wait for some modifications to then be active. So I know this is a little confusing, but so as I want to lead this up for a quick minute so you can see that um, there's two separate kinds of regulation, and they are on a case by case basis independent of one another, because you can have a gene that's always on, but that gene that's always on can either create an active enzyme or an enzyme that is then regulated in its function, right, after it's translated. Or you can have a regulated gene that's always active, so the increase in expression goes up, and as the enzyme numbers go up, they're always just active after translation. Or the last case is the double inducible, that you have to induce expression at a genetic level, and then the protein that comes off it is an apoenzyme, and then the modification occurs, which activates that enzyme, and then it proceeds to do its function. Okay, so that is that. All right. Okay. All right. So now that we've kind of got that out of the way, sorry about all that. I'm still getting back to using Zoom for this purpose. Um, there are two different post-translational uh, kind of um, reactions that can either activate or inactivate enzymes, and these are called allostery. So when we have enzymes, there are other sites in the enzyme that are not the active site, and these sites that can bind molecules that are not the active site are known as allosteric sites. And allosteric sites can either be inhibitory, meaning that the binding of a molecule to an inhibitory allosteric site changes the active site of the enzyme, preventing it from being able to do catalysis. The other is allosteric activation, which is the binding of an activator to a non-active site, again, an allosteric site. And this then causes a conformational change to the enzyme, opening up the active site and allowing the substrate to then bind. Right. So, and so how these actually work as activators and inhibitors typically is in some way, shape, or form um, done through the pathway that the enzyme is involved in. So in a lot of ways, if you think about this, feedback inhibition, right, or basically the creation of a product is what inhibits the function of the enzyme, makes a lot of sense, because if you're producing an enzyme that does a certain function, if you are creating a certain product, right? So here's the enzyme, it's interacting with its certain substrate as it forms its various um, intermediates and interacts with other enzymes along the biochemical pathway, you end up with an end product. And if you have a lot of substrate, but you only need a certain amount of end product, this end product goes to do, whatever. But once the end product has reached saturation, right, meaning that you've made enough end product that that you no longer need to make more end products, because you've met the threshold that you've needed, and now you have so much extra that it can't be used in whatever reaction system you're dealing with. The excess then binds to typically allosteric sites, and because you've made enough, you've made it in excess, right? The allosteric site says, okay, the cell has enough of this, and it needs to catch up. It needs to perform these biochemical reactions. And so this will continue to happen until the end product then is caught up from the cell. So again, you're going to have these reactions, and you're going to keep getting these products. And so as you keep getting these other products, so this end product is doing some other type of biochemical reaction. Once you've gone from this level down to this level, right? It's no longer at saturation. So the number of molecules available to produce inhibition goes away. So again, when this goes away, you're then back to active site normalcy, and then the substrates that the cell are taking in can then go down the product again to then feed back into the second system. So a lot of biochemical pathways, these end products and feedback inhibition, and there are some situations where you have something called feed forward, right? So you have feed forward activation. But by and large, again, going back to this whole like 80-90% of systems in biology are regulated through feedback inhibition, because once you've had enough of something, you don't want to keep spending energy on that thing. Once you have enough of whatever it is you need, you now need to move your ATP and move your, you have to delegate where you have energy. You have a certain amount of resources, a certain amount of energy. So you need to delegate where all that energy goes. So it just makes sense that if you have enough of something, you want to stop producing it until you need it again. And in some cases, we talked about, the different regulation at the genetic level, different regulation at the post-translational level. And a lot of these end products, take tryptophan, for example, actually do both. They actually do feedback inhibition of enzyme activity. So again, tryptophan binds to enzyme 1 way upstream in the pathway. And again, if enzyme 1 is inhibited, enzyme 2 never receives its intermediate, and then the system just shuts down at a protein level. But at the same time, if we have all these enzymes and we have these enzymes in excess because we no longer need this system, we also want to stop making protein. We want to, because again, that's also more energy. We're spending energy to create all of these enzymes, in this case, to make tryptophan when we have enough tryptophan. So tryptophan also exists to block, regulate. It actually inhibits regulation of the tryptophan output. And so in this case, we get feedback inhibition of the inducer or the regulator of the DNA that's making RNA. So whatever the DNA transcription factor is, right, the factor that binds the DNA to activate expression of these genes, tryptophan binds to it, and it prevents it from binding DNA. So if this is our transcription factor, it binds tryptophan, and tryptophan actually inhibits it from binding the trip operon, and therefore we're no longer making more proteins involved in the tryptophan biosynthesis pathway, and we're inhibiting whatever proteins we have created. And the best thing of all about this is that it requires no energy to stop using energy. So not only are we saving energy by not making more substrate or more product that we don't need, we're also not spending energy creating proteins that we also don't need. And we're doing it without spending energy. So now all that ATP that was being used for this, it now can be shuttled to a different system. So biochemical pathways in and of themselves, they come in different forms. There are linear, cyclic, and so linear example, right? These are just make a product goes through an enzyme, makes another product, makes another product through another enzyme. And one example of this is glycolysis, right? So glucose needs glucose hexokinase. Glucose hexokinase creates kind of that linear form of glucose. So glucose 6-phosphate, right? So it's kinase, so it actually creates glucose 6-phosphate. And so and so and that goes on and on and on, creating alpha ketoglutarate and phosphoglycerate, and all of those. And so every intermediate that's created from every different enzyme along that pathway, and then to an end product, right? And that's the key here. There's always an end product to a linear enzyme pathway. There are cyclic, right? And this is the movement of a particular intermediate into a system where it's then chemically converted by another enzyme, and then again the final byproduct, the final intermediate, can be chemically converted to that first intermediate that enters into the cycle. And so you get this cyclical chemical conversion and excuse me, use of those substrates in creating more products. And then finally we have branched multi-enzyme systems can be divergent. So this is, again, creating a product. This product is usually hydrolyzed into two other products, and then those two products are met by two separate enzymes, and they undergo different biochemical pathways. And then there are convergent ones, right? So this is a great example of amino acids, because again, amino acids or even nucleic acids, right? There's the amino group, and then there's the R group, and they exist typically in two different uh, places, right? So we can make the amino group and the R group, and then we can combine all of those elements into those groups, and then we take those two groups. So let's say this is the R group and this is the amino group, and then we combine them to create the amino acids, and then they can go forward and yeah, be modified from there. So I've talked a lot about um, all these factors that affect enzyme activity, and a lot of these things that and from the perspective of the fact that we've talked about it in lab, and also I've been very kind of adamant about the fact that the environment in which a protein is made, all the different factors that are in that environment, all the things that are around and interact with that protein as it's folding, affect its folding, and that's how specific the enzyme that folds, or any protein that folds, and how any modification from any of those, or any deviation from any of those specific environmental conditions, can result in a conformational change. And therefore, and again, if you have a change in conformation, or in other words, a change in shape, you have an effect on activity. So some things make a lot of sense, right? So temperature, water concentration, pH, the presence of inhibitors, right? End products which can act as inhibitors or activators, which we just talked about. And then we talked about how the amount of substrate can affect the activity, also the amount of protein that's made, and obviously how much enzyme you have available to you does affect how quickly or how much of that chemical reaction you can do, right? And the last one here, we talk a lot about this with radiation, and this is because especially ionizing radiation, just as the name suggests, can ionize atoms and charged particles, which means that if you have particles that are charged, or even particles that are uncharged, if enough radiation hits those molecules, it can send a lot of the electrons in those atoms into a different energy state, and that different energy state can change the actual chemical composition, or at least isotype uh, uh, of those particular atoms, and that can affect their ability to do chemistry. All right. So the last 10 slides or so that we have in this is how kind of all of this plays into defining different features of microbes, and based on their ability to perform central metabolism, and coupled to just general environmental conditions that are involved in their ability for their enzymes to function. So the first one is obviously kind of this one, presence of O2 and the ability to grow in O2 really is a defining feature of kind of the environment that that these bacteria live in relationship to the type of central metabolism that they can undergo, whether it be fermentation, oxidative or anoxic, oxidative phosphorylation, or anoxic, or anaerobic, excuse me, that's what I'm looking for, or anaerobic respiration, which still yields same number of ATP. But again, having the electron transport chain and having the drivers of the electron transport chain, like a terminal electron acceptor, are all really important here as far as the defining environment in which they live and what they can use to drive those different systems. So the first one, talk about our obligate aerobes. These are aerobic respirators. None of them can perform fermentation, we are obligate aerobes, and you might be thinking, okay, well, you talked to us about how muscles can ferment, and make lactate and lactic acid when they're deprived of oxygen. That's true, but when they do that, they're not they're fermenting because they don't have access to oxygen. And of course, when you have lactic acid buildup, you have pain because you're actually having some trauma to those muscles. So the buildup of lactic acid and performing fermentation is something that can happen, but it does cause damage to those cells. And same thing with obligate aerobes. If you take away oxygen, they could probably survive for a subtle amount of time, but eventually over time they would die. Just like if you were to deprive your entire body of oxygen for a long period of time, you would also die. So obligate aerobes, examples: Mycobacterium tuberculosis, Pseudomonas aeruginosa. This should make really good sense because both of these infect the lungs where you have a high amount of oxygen. Microaerophiles, these are uh, bacteria that can grow under aerobic conditions, but only small amounts. So the oxygen level has to be between 2 and 10% of what atmospheric oxygen is, which is actually at 20%, because 70% of atmospheric air is actually nitrogen, right? So these require a really kind of small amount of oxygen, um, but they still perform aerobic respiration with a small amount of oxygen. But too much oxygen is actually toxic to their enzymes, meaning that there's a certain amount of oxygen that they need to just drive that aerobic respiration, but they don't need any more. And one example of this is Helicobacter pylori. This is, again, makes sense. This is something that lives in the stomach of mammals, especially humans, and it actually, inside the stomach, and inside like the fundus and where the acids are, there is still some oxygen because the cells in that area do need to be able to get access to oxygen because, again, all of your cells are obligate aerobes. But most of the oxygen in that area are absorbed by the cells, leaving very little oxygen left in the large vast majority of the stomach. The next are facultative anaerobes. These are some of the most diverse and I would say biologically fit organisms on the planet because they can kind of oscillate between both environments and be successful in both. So they basically grow aerobically when oxygen is available, and then when oxygen is deplete, they do have genes available to them to undergo anaerobic respiration. So these are typically non-fermenters, meaning that only under certain extreme conditions will they ferment just to stay alive, but they have a lot of biological flexibility in that they are really good at growing aerobically, and performing aerobic respiration and oxidative phosphorylation, but that they can then drive anaerobic respiration and still generating this 36 ATP in the absence of oxygen. There are aerotolerant anaerobes. So examples of this are E. coli and Salmonella. This makes sense because they live both on the outside world, like outside of shell eggs, and outside in the produce, and all these other places in the environment. But they also live predominantly in the gastrointestinal tracts where there's very little oxygen in mammals and birds and other types of animals. They're aerotolerant anaerobes. These are typically, again, bacteria that grow more successfully anaerobically. They usually are defined by their ability to either ferment or anaerobically respire, depending on what's available to them and their gene setup. And they can grow in the presence of oxygen, but they don't utilize oxygen. So means that oxygen isn't toxic to them, but they don't use oxidative phosphorylation. They use other non-oxygen versions of terminal electron acceptors. And two good examples of this are the streptococci. So strep pneumo and strep pyogenes, they live in the throat and in the upper respiratory system, which still gets a lot of oxygen because you're breathing in a lot of oxygen. But as you breathe in, most of the oxygen you are getting is filtering down to your bronchi and your alveoli of your lungs and being distributed through your body, meaning that the amount of oxygen that they can get access to is very little, and that's fine by them. They don't mind. But the presence of oxygen coming by them also doesn't hurt them. And this is an extreme difference to obligate anaerobes. These are, again, fermenter, just like aerotolerant anaerobes. These are predominantly fermenters. Some of the faster growing strains or species are anaerobic respiration. This is because oxygen is actually lethal to these. And also oxygen and oxidative species are lethal to these bacteria. And this is for a number of different reasons, but a lot of it has to do with the fact that oxygen can kind of replace certain parts of their biochemical pathways that are already set up to engage with anaerobic and like things like anaerobic terminal electron acceptors, etc. And so when oxygen replaces those enzymes cannot bind oxygen, and therefore those enzymes no longer work, and then they can't create ATP, and then they die. So as much as oxygen is involved in the creation of ATP, the next kind of set of things have to do with like this concept of optimal growth rate, which is conditions in which the enzymes inside of a bacteria, so all the enzymes collectively, work at their highest efficiency. Therefore, the growth rate is at its peak amount. So this isn't about where the highest number of cells are. This is the most number of cell divisions. So the optimal growth rate is equivalent to what conditions exist for the number of cell divisions to be at its maximum. So the first thing that we're going to talk about is temperature. Temperature obviously affects, like we showed you before, it affects enzyme functionality and the ability for cells to grow in certain conditions, certain temperature ranges, to kind of defines them in a classification scheme. We have psychrophiles, which grow in they can grow from sub-0 degrees centigrade all the way up to 20 degrees centigrade. So this is room temp to kind of to freezing water. There are also extreme psychrophiles, and they grow way out here in subarctic temperatures. So you can find these in like frozen lakes and the Arctic, Antarctic shelves. There are mesophiles. These are typically cells that grow from room temperature to internal body temperature of humans and most mammals, which is about 37 degrees, 38 degrees centigrade, which is about 98.6 degrees, maybe up to 100 degrees Fahrenheit. Above this, we have then thermophiles and hypothermophiles, and these start getting into temperature ranges which start moving towards, hot springs and then, sulfuric or hydrothermal vents underneath the ocean. We have pH. Those that can grow in acid are known as acidophiles. Those that grow in neutral pH are neutrophiles, and then those that grow in basic pH are known as alkalophiles. We also have osmotic synthesis, right? Osmotic regulation. There are halophiles. Halophiles are bacteria that grow in high salt concentrations, and then there are osmophiles, which grow in high water concentrations. And you can have the combination between the two, osmohalophile. And good examples of these are Vibrio species. which live in the ocean. So they're surrounded by water and they're surrounded by salt. So, as it relates to growth, enzymatic function really has the important part about, uh, kind of twofold, right? There's one aspect of this that has to do with the fact that you need cells to grow as a cell in order to divide, because again, reproduction in whatever form that takes, reproduction is a consequence of biological fitness, right? If you're biologically fit, as Darwin puts it, you should be able to reproduce. You should be able to get to an age that and have enough resources to reproduce. And for bacteria, this is true. And so the base kind of tenet here is that physiology, or central physiology, does allow for cells to grow because again, they have competed for the resources in order to grow. And the whole point is that because they've been able to acquiesce resources, they can create ATP, and this then results in enzymatic functionality. First, to survive and again exist in prevalence, they exist, and then have an excess or an abundance of that ATP in order to drive uh, growth and division, which all depends on enzymatic activity. Everything from separating the cell, making the septal ring and creating more phospholipids for the membrane, more peptide glycan for the cell wall, all the way down to replicating DNA, because you need to replicate DNA to make two daughter cells. So last couple slides, have to do with just kind of how physiology has to do with growth and how growth kind of uh, exists in kind of a really important two different fold functionality of physiology. So bacteria exist more or less in two distinct states. There are free swimming bacteria known as modal bacteria or forgetting the word, not sessile. Uh, Sorry, I can't think of the word. So you have free swimming bacteria, bacteria that are out and about, and they're either swimming by nature of their flagella, or they are swimming in the absence of flagella by movement of their environment, right? So these are out and about, and they're moving. They are still thinking of the word, but I can't think of it. But anyway, uh, once they get to a point where they then need to start to colonize a certain environment, because again, you're going to swim around until you find something that you like, and once you have found an environment in which meets your resource and kind of protection kind of situation, um, bacteria can then attach themselves to surfaces. And once this happens, bacteria convert from being free swimming to something called sessile. So we go from free swimming to sessile. Um, and then sessile bacteria then start to shed their capsule. So as these bacteria have their capsule starts to expand. So they create more capsule, and so the capsule that starts like so, that's attached to the cell, gets pushed out for a new capsule, and so you get this layering of capsule. And this layering of capsule creates what we call the slime layer. And the slime layer continues to grow out. And this then creates the first iteration of irreversible attachment and proliferation, plus the creation of their slime layer. Both of these then combine to create what is known as a biofilm. And the biofilm is basically the same glycocalyc structure as the as a capsule, but the capsule that has been expanded out into space. Now, in the biofilm, biofilms begin kind of in a two-dimensional structure, and then they can grow into these three-dimensional structures, which house a lot of bacteria. And the inside the biofilm only has a certain carrying capacity, meaning that all the nutrients flowing through it, having access to whatever is attached, and the stability of that biofilm attached to a certain surface, all are factors in the maturity of a biofilm. But once the biofilm has started to reach its carrying capacity, there's not enough resources or attachability places to go around, and cells start to die, or cells can't find a place to attach. The bacteria that can't attach but are still alive then switch back to this modal modality. They go back to free swimming modality, and where they can find a new place of attachment that is, again, rich with resources and hasn't been tapped for their resources in order to generate another biofilm. Most canonical scientists will tell you that 80 to 90% of bacteria in the world at any given moment exist in a biofilm. And this is because biofilms are very protective. Then they also allow for the bacteria to colonize and again persist, to exist, to get access to resources and compete for resources and be protected. And so part of this protection is that the biofilm itself is actually pretty charged, and this is because of the types of sugars. As they break down or they dissolve over time, they start to break apart and leaving open hydroxyl groups and oxygen groups, and giving this generation of negatively charged calyx on the outer ridges of the biofilm. And this allows the bacteria to kind of exist in all parts of the biofilm. And this gives a kind of a separation or a gradient of life inside of the cell, in the sense that there are cells that are more to the cortex, or the center part of the biofilm, and these cells are really involved in cell signaling, regulating cell death cascades, having changes in their relative physiology to survive, and typically very quickly become persister cells because they have less access to nutrients. So the creation of persister cells, and as these cells start to die, or regulation from the inter parts of the biofilm, allow for cells to signal to one another, especially to the fast growers or to any diversity that is generated inside of the biofilm. The relative health and stability of the biofilm. Is a biofilm getting too close to the end of its carrying capacity? Is it getting too full? What are the requirements for the cells and all the biofilms to survive? So the other really important thing about the charged part of the nature of this biofilm is that because it's so charged, any nutrients that come through get trapped in here, as well as any antimicrobials, especially those that are charged, like defensins or like complement. These things that are charged can't penetrate through the biofilm, and because they get stuck and caught up in these negatively charged particles, they never reach the cells inside it. So biofilms, by and large, they have these localized gradients in different parts of the biofilm, and this allows for kind of the creation of a community of cells. So typically a biofilm will start as a homogeneous species, but as the biofilm grows, especially in nature or even inside of a human or wherever it is, bacteria are surrounded by other bacteria. And so how the biofilm actually gets structured really depends on the type of cells and their ability to grow. So go back to those other things: pH, availability of nutrients, availability of oxygen, right? All of these things happen in a gradient. So as you move toward the outside to the inside of a biofilm, there's more of everything: oxygen, nutrients, pH balance, on the outside edges of the biofilm. Um, and so, you have the situation where you'll have cells that can be involved in resource capture. You have cells that can uh, cooperate with one another, right? Especially so, like, if these outside uh, cells are really good at metabolizing starches, the ones on the inside might be really good at metabolizing glucose. So these guys get the carbon source they need. These guys get the carbon source they need, and this helps keep most of the cells healthy and strong, and being able to exist along those gradient lines based on their metabolic strategies. So here's just kind of a view of this, right? So O2 is on a gradient, nutrients are on a gradient, pH is on a gradient. Um, and same thing with, and so these are all coming from the outside to the inside. Now, because most of the cells exist on the inside, the quorum sensing gradient actually kind of goes in reverse. So the ability for bacteria to speak to one another actually is at highest density inside the biofilm, and it gets progressively more diffuse as you move outside of the biofilm. So biofilms, just kind of in general, are really important for cellular communication, cooperation, both from a metabolic standpoint as well as from a survival standpoint, right? Um, and the ability to coexist, to either make molecules that other bacteria need that you don't need, so that they can give you something in return, or some other type of synergy in their ability to get access to those molecules. And then, again, you're going to have some competition in the biofilm. And this is kind of the part of the biofilm that actually makes some sense, but not a lot of sense. So competition is important from the sense that only the strongest survive. If you exist in a multi-species biofilm, you want the strongest bacteria that are going to help you survive, right? And if you have a bacteria that isn't strong enough to withstand a certain antibiotic or a certain nutrient depletion event or something like that, you want those cells to die, because again, all the cells in a biofilm are contributing to that biofilm. Just like a house of cards. If you pull one of those cards out because a certain species dies, because they couldn't cut it, then the whole house of cards collapses. And same thing with a biofilm. So there's always this constant cooperation, both metabolic and survival cooperation, usually inducible through chemical and electrical communication. So the cells talk to one another to produce toxins, to not produce toxins, to digest certain things, make exo and endo enzymes, to have this like harmonious genetic response. So there's an increase for survival for all species involved, but to ensure the stability and survival of the biofilm, there's always competition, in order to make sure that the biofilm only has the strongest species available. All right, I know that's about 15 minutes longer than I had hoped, but that finishes metabolism. If you have any questions, please reach out to me. I'll also be posting the homework for this to be done after break. If you're reading this during break, I hope you have a great break. If you see it after break, I hope you had a good break. All right, I'll see you in class after spring break.